Hello, thank you for joining me today. My name is Isabel and I want to take this opportunity to talk to you about some of the research that I've been carrying out here in Cambridge over the last couple of years. I work on a group of organisms known as Dinoflagellates, and so I think the most sensible place to start today is with a bit of an introduction to what a dinoflagellate is. Dinoflagellates are a group of related species. They all live as single cells. Most of them are found living in water, and again the majority of them make their food from sunlight by photosynthesis in exactly the same way as land plants do. Some of them live swimming about by themselves, and some of them live inside other organisms, and these are perhaps the best known dinoflagellates and the ones I wish to mainly talk about today. So this is a picture of one of the coral reefs, and this is probably the place where you would find the most famous groups of dinoflagellates across the world. So what we're actually looking at here is the product of two species working together. You have the coral polyps, which are responsible for building the hard structure that makes up what we normally think of as the coral reef. But living within those coral polyps are dinoflagellates, a completely different species, and these two species work together in a beneficial relationship. The coral gets a supply of food from the dinoflagellates that they are in turn making by photosynthesis, and the dinoflagellates get a nice, safe, stable, protected home to live in, much more convenient for them to live inside this hard protective structure that the polyp has made than to live swimming about in the ocean where they are prone to being eaten by other species. Now sadly, the place we mostly hear discussions of coral these days is in the context of coral bleaching. Coral bleaching occurs when sea temperatures rise, which causes the polyps to lose their dinoflagellate partners, and then those polyps go on to die without them. And so you get this change over really just a few months from the beautiful colourful structures of live coral and dinoflagellates on the left to the bleached and dead structures that we see on the right of this image. We know that one of the first stages of bleaching is the loss of dinoflagellates. We think that part of this is dinoflagellates being damaged by the rise in temperature, but we're quite unsure about exactly how that happens. Why are dinoflagellates so susceptible to relatively small changes in temperature? We have some theories, but a problem for dinoflagellate research over the last few years has been the lack of tools to properly investigate them. And that leads quite neatly onto what I've been working on for the last few years, which is trying to develop a new technique to allow us to look at what's happening to the dinoflagellates in more detail. Scientists have been trying to develop tools to allow us to put new genes into dinoflagellates for a bit over 20 years. Now, a lot of the theories about how bleaching works could be confirmed by putting in new genes which would help the dinoflagellates resist higher temperature. If that new gene makes a difference, then that would be a sign that the old gene was a part of what makes dinoflagellates so vulnerable to these rises in temperature. And so a lot of what I have been doing over the last few years has been trying a range of different approaches to try and get new genes into the dinoflagellates. One of the problems that we have been encountering in this process is that dinoflagellates are really very tough. Designing the DNA that we want to put into them has been relatively simple, but getting that DNA inside the cell where it can start working has proved much harder. So we have ended up using an old technique which goes by the somewhat odd name of Biolistics. It involves taking tiny gold bullets, much smaller than you would be able to see without a microscope, coating those bullets in the modified DNA that we're looking to put in, and then shooting them at the dinoflagellates. So what we end up with is some of these gold bullets lodged inside the dinoflagellate cells themselves, and that's how we've ended up transferring the DNA into the cells. I explained this process to my soon-to-be mother-in-law over lunch a few months ago, and she gave me this picture a couple of weeks later. And while our gun doesn't quite look like this, it's not too far from the truth of how I spend some of my working days. As we developed this tool, we started out with 2 different genes which would give us a nice clear signal that we'd managed to get our new DNA in. One of the things that we tried putting into our dinoflagellates was a form of drug resistance, and you can see that on the left of this slide there's nothing moving, but on the right you can see little oval blobs moving around. And what we're actually looking at here is the view down a microscope, and those oval blobs are dinoflagellates swimming about happily. The DNA has given them a gene, making them resistant to our drug, and so they're still alive. While the picture on the left is of the dinoflagellates we didn't give this new gene to, and so they have all died. More attractively, we've been putting in a gene which allows us to stain the dinoflagellates blue. So on the left again, we have normal cells which we're unable to stain, and so they show up as a red colour under a fluorescence microscope. But on the right, we have the modified cells which we can stain blue. And as my nephew is very fond of telling me, the red and blue together gives him a pink finger painting and gives us these pinky purple looking cells. And so these 2 experiments together let us know that we have been able to put new genes into these dinoflagellates and have those genes work the way that we were hoping. So now that we have this new tool for our toolbox, what's next? Well, over the last few months we've been sharing this technique with other labs around the world. This lets these groups start trying to look in more detail about what is going wrong inside dinoflagellates during bleaching. Some of the things that my lab are planning to look at are putting new genes into dinoflagellates which will signal us when certain processes get disrupted inside the cell. Another project that we're starting is putting in modified forms of some normal dinoflagellate genes which we think will then make them more resistant to temperature rises. If that ends up working, then that tells us that we're on the right track with our theories about what is causing bleaching. Now, none of this will solve the root problem, which is rising ocean temperatures, but it will help us to understand what is going wrong for the reefs in a little bit more detail, and that's a key step in trying to help them. That leaves me with one remaining task today, which is saying thank you to actually quite a lot of people. First, I would like to thank the Gordon and Betty Moore Foundation, who are the organisation who fund this research here in Cambridge. I'd also like to thank the rest of the lab where I work, and you can see us all keeping our distance from one another while carrying on some of our work. Another thank you is to the Botanic Garden and the Cam Plants Hub for hosting me today, and finally to all of you for taking the time to let me explain some of the things I've been working on recently. So thank you very much, and enjoy the rest of your day.